First, as Ginger and Eddie kind of said, it's really hard to predict the future. If you look at past predictions of the future, where, say, in 2001 we were supposed to have, hotels orbiting the Earth and that sort of thing, and that wasn't written that much before 2001. So take all predictions with a great deal of salt. And maybe it'll come back. Okay. So I thought I would talk about some what might be considered timeless questions in microbial ecology, since I was supposed to talk about the future of microbial ecology. And I thought I'd ask these sort of questions, like fundamental characteristics of microbial communities and how relevant they are. And what are these characteristics? These are things we've seen a lot of today. The composition, and in fact, here's something we actually heard Jennifer Brum talk about, was the composition. We want to try to determine it in ecologically relevant units. What are the functions? How do these organisms connect? And we've seen a lot about that. How do they interact? And a lot of microbial ecology also includes knowing rates of these processes, which are arrows in various diagrams. I would just show a food web diagram up there, but they're arrows in redox cycles and in food webs and all over the place. And it's also really important to consider that microbial ecology happens at multiple scales. You have molecular scale, what's going on inside of cells and between molecules. You have individual cells and how they interact with each other, positively or negatively. You have mesocosms, which are the kind of sizes that we tend to study, bottle sizes of samples. And then you have a bigger community, and you have ecosystems, and then you have global. And microbial ecologists actually try to study things at all those levels. So integrating across the levels is one of the big important things. And Tara has done an incredibly good job of trying to integrate the global measurements from individual bottles that they looked at. So I was going to talk about how the newer 21st century approaches are going to address these questions. And I'm not going to go through this whole slide because we have other things we'd rather do. And other people talked about history, but I tried to sort of put the last 50 years of microbial ecology and some of the important accomplishments that have happened, and you can glance at it, probably read it faster than I could. In the 70s, high abundance in biomass of marine bacteria was found by epifluorescence microscopy. The 80s were really important, I think, in microbial ecology, maybe because I was just new in the field or something, but the growth rates were measured, and it showed bacteria consume about half of the global productivity. And I think that one point made a connection between everyone else that was looking at the world and these microbiologists that were studying microbes that seemed to be irrelevant to most of the rest of the world before then, except for disease or something like that. Suddenly, if they were eating half the carbon, now every department had to hire somebody. And in fact, a huge number of microbial ecologists got jobs because people said, okay, they're not just somewhere eating garbage and fish poop on the seafloor. We have to actually put them in our models and figure out that they're part of the system. So I think that was really, really important. People focused on material cycling and quantification of rates and not who was there. There wasn't very much of that in microbial ecology, except the phytoplankton ecologists always throughout this time had been studying the recognizable ones. Now, late in the 80s, Norm Pace and his colleagues found you could clone and sequence microbial small subunit ribosomal RNA, and the concept of extracting DNA from nature and getting sequences out of that suddenly became a great new idea without cultivation. And right around that time, PCR was developed. That was a really important thing. High abundance of viruses was discovered in the late 1980s. Evidence of high viral infection of bacteria and phytoplankton. As we just heard from Ginger, flow cytometry came into the field. Then Prochlorococcus was found to be the most abundant phytoplankton in the ocean. And people started to integrate microbiology with other oceanographic studies, and I think that was a really big deal. So all that stuff happened in the 80s. It was a great time to be in the field. In the 90s, RNA cloning and sequencing with PCR was really popular, and people had what I called the golden area of finding new phyla, and I could tell you literally every sample you looked at, you'd find a new phylum. It was that easy. It was like what they used to call Hershey heaven in microbiology. Do the same experiment over and over and over again and get a brand new exciting result. And it actually ruined a lot of the creativity in the field because it was so easy to get exciting new results. And I actually think in some ways there was a detriment in terms of creativity. We also found that there were non-extremophile archaea. Archaea were thought to only be weird things that lived off in some strange hot springs and stuff. Turns out they are one of the most abundant bacteria type organism in the whole world, in the ocean. Some expansion of environmental genomics started to happen beside ribosomal RNA, and we started to see other sequences. And people looked for applications of process and rate study, but diversity was really hot in the 90s. And in the 2000s, sequencing got cheaper. Genomics and metagenomics came in because people were able to now, with the cost going down, able to get other genes. Proteorhodopsin was discovered by the DeLong and his colleagues, and that was really neat, although it's interesting, we really don't know what it's doing in the system today, even though that was a long time ago that they discovered it. Clone-free amplicon and fragment sequencing, that was actually a big deal. If you could sequence without cloning, there was a whole bunch of biases in cloning that people didn't even know about. And by doing clone-free work, those went away. 454 and Illumina work. Global Ocean Survey, we haven't heard much about that, but that was actually a big deal. Okay? Genomes were assembled from metagenomes early on. Tyson and Banfield and others started it, and then other people started to do it. Molecular knowledge became de rigueur for microbial ecology graduate students. They all had to know how to be gene jockeys starting in the 2000s, and a lot of them forgot how to do like counting things and going to sea and measuring stuff in field. They all thought, oh, I just need to study genes and I'll understand everything in the world. Okay? In the 2010s, cheaper sequencing happened, and there's all optical and other methods. TARA studies we saw today, incredible stuff. And still, not much new on rates. The rates that we did in the 80s, by the way, are still the same rates that people do today. There has not much been done, even though that if you look, it's an incredibly important part of ecology. Okay, but I won't talk about rates very much. Anyway, I already did. So, composition of microbial community, and what are the relevant units of diversity? Fundamental units of diversity is organisms and their abundance, what kind they are, and you should resolve organisms into some kind of units, then not mix them up, and they should have some kind of coherent ecological characteristics. In other words, if this organism behaves differently in nature than this organism, you would like to keep them separate, right? I think everyone would agree with that. Okay? We usually don't do that. We usually have some practical way to separate them, like a % difference of a molecule or something like that, or what's easy to do because it's really hard to do it this way. But in reality, we want to separate things that are different that we might call ecological species. Okay, so how do you do that? Well, you have to kind of think how people look at organisms. We often talk about type species or strains or reference genomes, and they're by definition clonal. They're all identical. Okay? How representative are these identical organisms to their wild relatives? Does it matter? And to what extent are natural communities clonal? Is a really good question. And do patterns differ by taxa? Is this group mostly all the same type and this other group totally diverse? Okay? Now, when we think about clonals and cultures and stuff, I think it's important to keep in mind that if you have something growing in the lab, it's really cushy in there, and it has no need to defend itself against anything. You're feeding it, you've protected it against viruses and protists and immune systems, and they generally lose those things. Okay? Now, in nature, these things that kill them turn out to be just as important to their survival as what they grow on. Okay? If something kills you, it's too bad, you're dead. Okay? It doesn't matter if you grew faster than the other guy, because you were killed. Okay. So avoiding being killed is as important as growing fast. We never think about that. We think about growing fast, because we're thinking of cultures. And in nature, there's a lot of trade-offs between growing fast and defending yourself about being killed. And these are things I think we need to consider because they involve diversification of organisms. Okay, now let's talk about how highly resolving methods might work. You need to resolve things really well. And in fact, if you have the choice of resolving too much or too little, always choose too much. Because you could aggregate your data if they're too resolved into what you want. But if your measurement is too coarse, you can't unresolve it. You know, you can't redefine. Now, I'll draw an analogy to colors because I think it's really easy. Okay, if you discretely sample a rainbow, you'll think that the rainbow is discrete. If you pick a yellow and a blue and a purple and a green, you'll think, the rainbow is just these 5 things because you have a small sampling. Now let's imagine that diversity looks perhaps in some organisms like this, they're really fairly discrete. In other organisms, totally continuous, like they're all different from each other, or someplace where there's a lot of them and there's a lot of fine resolution there. Now, if you didn't have a really high sampling frequency, how could you tell this from this? Answer is you can't. Okay? So I think you need to have good resolution in your measurements and a lot of sampling frequency to really figure this out. Okay. So why do we care? Well, Closely related organisms we know from the few cultures that we looked at, they tend to differ in islands. And the islands very often are things that are resistant to viruses or grazing or host immunity. Sometimes they're for competition because they have some special advantage. Now, variations always carry some benefits and some costs depending on what's happening. And years ago, Hutchinson, G. Evelyn Hutchinson, talked about the paradox of the plankton, where I think it's still a valuable concept. He said, if the plankton are all competing for a few resources, why is it that one or two types don't win? Okay, why are there only not a few types? And he knew there was a lot of different kinds of phytoplankton. And people said, well, population genetic theory and competitive exclusion theory said somebody should win. And this was an old question in ecology. There should be winners and losers, and the losers should go extinct. And people realized that one of the main answers was, well, there's patchiness of the conditions. Things are never the same, and these theories only work under constant conditions, okay? And small differences in fitness don't necessarily stay for a long time. So these things are patchiness both in biological and physical chemical environment. Remember, biological environment is as important as the physical environment for organisms. So also, in addition, organisms can partition resources, and we've learned a lot about that. And one thing about this that Hutchinson didn't talk about is the death side. There's selective infection and there's selective grazing. And that's why certain kinds survive and certain kinds don't, okay? So all these things help the dominance stick around. And how does it work and what are the adaptations? And that leads us back to clonality in nature, because if you take a look at what metagenomic data tell us, you said, well, we really couldn't tell much about this until really recently when we could really sample a lot because sequencing is cheap enough. Okay? So it's really pretty much only the 21st century level of sampling and sequencing that we can start looking at this. So, has any studies looked at it? And as Ginger talked about, her former advisor, Penny Chisholm, someone has looked, and Chisholm's lab has looked really closely at Prochlorococcus. And this paper was published in Science last year, and it's really quite remarkable. It's a great model organism, partly because it's like one of the most abundant organisms on Earth too. It's not just a model in sort of generic sense. What's interesting is these are, by the way, all highlight 1 Prochlorococcus. In the work that we saw today, it's 1 OTU. Keep that in mind. This is 1 OTU in almost everybody's studies. Now, they looked at 90, and they're also more than 98% identical in the intergenic spacer region, not just the 16S RNA, but the spacer that's highly variable. They looked at 96 different single-cell genomes from the Sargasso Sea, and they found the genomes were different. And that they could use the ITS sequences to break them up into clusters, and these are genomic sequences using to separate them, and they're quite coherent with each other. And they concluded that this one population, this 1 OTU, consists of hundreds of coexisting subpopulations, each with what they thought was a moderately stable backbone of core gene alleles. In other words, different varieties of the same gene. They all annotate the same, but if you look at the sequence, they're different. And they had different sets of flexible genes between these clusters, okay? Now, here's what's interesting. These diverged millions of years ago, and they have been stably living in their own little units as separate entities for millions of years side by side in the ocean. Okay? Now that should be ecological species, right? They are stable for millions of years and they have different sets of genes, and we are usually lumping them all into one category. Okay? So fine scale matters, resolution beyond most of the current studies is really meaningful. Okay? We don't know why some of these things are better than others and under some conditions, but I think for the 21st century, I think we need to start moving to these kinds of levels of resolution, not what we could kind of do. Now, my postdoc, Nathan Algren, was looking at our time series station at these particular Prochlorococcus, highlight 1. This is the percent of all the cyanobacteria that are in this highlight 1, and this is 4 years of data monthly, and these are 4 different varieties of it. And 2 of them are negatively correlated to each other. When one goes up, the other goes down, okay? So we can find at our site that these guys have ecology. They're different from each other, and these vary only in a few bases in the ITS region. Otherwise, they're virtually identical sequences. The 16S is the same. Okay, so I predict that we will, this by the way is this is the crystal ball here. This means I'm just gonna be totally wildly speculating. And I predict that we will expand these to a lot of different microbial taxa in coming years. and we'll look at fine scale differences of things that we now call one kind of thing. And I think that when we heard from Tara and others that there's 35,000 OTUs, I think that's way too low. I think there's probably millions of different things in the ocean that are different, that behave different from each other, okay? I predict some patterns will resemble Prochlorococcus and others won't. And it'll be really interesting to see why. We expect some organisms which have kind of boom and bust life, like after a phytoplankton bloom, they take off. You kind of expect them to have less diversity than the sort of slow and steady ones, but maybe that won't always be the pattern. And we have preliminary data from SAR11 and marine Taumarchia saying they have a lot of closely related variants, and some other common taxa like the actinobacteria don't seem to do that. And as with Prochlorococcus, the variation seems to have meaning. This is some work from my student David Needham, and this is time series from our San Pedro Ocean Time Series also, like Nathan showed, halfway between Los Angeles and Catalina. And this is samples between March and August. There's 43 samples. Some of them were done every day for a few weeks, and some were every week. Don't worry about the details. These are SAR11 ITS OTUs. In other words, just the ITS sequence from SAR11, highly variable sequence. These are 99% identical. 99% OTUs, so they're 1% different from each other. There's 20 different ones from SAR11. If you take this one 99% OTU and look for one base difference in the ITS, one particular base, here and there, you find that there are 7 types that have one base different from each other. And then you look over the year, look at this. These 2 types were really common early during the spring bloom, and this type was common in the summertime when it was oligotrophic. So these types that differ by one base in the ITS are different ecologically. So I think there's a lot more detail in these systems that we aren't seeing yet, unless we start looking really closely. This is real ecology, okay? And just to show you, Tara data have this too. Nathan looked yesterday in the Tara data, and he's really quick, okay? And he mapped the distribution of Taumarchia at the DCM using metagenomic reads and using competitive recruitment to these genomes. And these are the winners of the competitive recruitment, and you see different ones in different places. This red one is one that we have a culture and enrichment. Interestingly, it was the most abundant off where we enriched it, which is always nice to know. And oddly enough, look at this. It's also most abundant off South Africa. Why is that? It's not in between here. Everything's everywhere, and this is where it's selective. I don't know. Anyway, I'm not an everywhere is everything guy. Everything is everywhere, but it's very interesting. And the Tara data, we already heard people are downloading the data and playing with it. Yeah, right now as we speak, there's like 50 graduate students and postdocs busily doing all kinds of great stuff with the data that you never even thought about. And you're going to get cited right and left from these people. It's great stuff. Okay. So now here's some other general predictions. This is a real easy one, okay? There's going to be a greatly increased reliance on cheap sequencing, informatics, and automated data analysis. Much less reliance on people actually looking at samples and on their intuition. Our data sets will get bigger and bigger, and there will be so many sequences that we will be chronically short of computing power. I think that's a duh, this one. However, data sets require really thoughtful reduction and filtering, and we know that unexpected results often represent new discoveries when human beings look at data, okay? And thinking outside the box. Computers have trouble thinking outside the box unless you tell it to look outside the box. So you have to be really careful of relegating all your analysis and having it all unsupervised. Be really careful. Unexpected discoveries could be lost. And there's probably a lot of stuff today which is, the 4th domain could be in someone's data set and they just didn't look at it because it didn't match something they were looking at, okay? And I think there's a good story I heard about Eugene Odom's graduate students. It was from Scott Nixon, who was one of his former graduate students. And he said that Eugene Odom would go into his students' offices and look in the trash cans. And all the interesting discoveries were in the trash because they did not meet expectations and they thought they were wrong. Okay, and if human beings think their data are wrong and throw them out, computers are even worse than that. They don't even know right and wrong, they just know what you told it to look for. So be really, really careful not to relegate everything to the computer. Try to look more at your data, look more openly at it, try to think outside the box. I think that's really important. Now, I think with increasing artificial intelligence, we could program computers to look for the unexpected and at least alert us I didn't think this was coming up, you know, should I throw this out or should we look at it? You know, it's like the spam folder. It says, look at this, is it really a message from your mother or is it spam? Okay, and some things are much more important than others and you should be alerted of these things. Okay, so moving along with my more predictions as shown by this gentleman here. Okay, a big problem in metagenomics, everyone knows metagenomics is really powerful, is that a large number of the sequences are unannotated. They sit in those big unknown boxes, okay? And there's too many of them. Only a fraction of genes can be annotated to function. And you know when the function says ammonium transport or peptide transporter or reductase, those are not useful at all, but we call those annotations. Okay? If we are a phytoplankton ecologist, we want to know what is the Km and the Vmax of that ammonium transporter. Because if it's micromolar, it's not working in the ocean. Okay, or something like that. So we need to know much better information than virtually all these metagenomic information is telling us. We want that stuff if we can get it. Now the problem is to get that, ideally you have someone studying in the laboratory and every single enzyme gets studied. We know that will never happen ever. We're lucky if 1/10 of 1% of the proteins will be studied in the laboratory. So here's my answer, my hopeful prediction. That, and this will take a long time, I think 20 to 50 years from now, our chemistry friends that study proteins will be modeling proteins. Now, proteins are just machines. They're little machines. And I think they're going to be able to model these machines, and just the way we work with mass spectra down to like the 12th decimal point or whatever they work with, they're going to figure out all the tolerances of these machines and figure out exactly what fits in this slot and how this could move here. And their computers, which are getting so incredibly powerful, are going to be modeling these proteins. And I predict that 50 years from now, we'll be able to look at a DNA sequence of a protein and a computer will say, here's the substrate, here's the range of substrates, here's the affinity. Or this protein interacts with that protein. Look, they're two subunits of the same thing, and I could predict that from scratch. I really hope that computers can do that because we'll never do it mechanically in the lab. Okay? And I think if we can do that, then microbial ecologists could use that information, because now suddenly really useful information will be available to us, like substrate specificity, affinities, and that sort of thing. And I think that microbial ecology will really depend on something like that. Okay? And might be able to match viruses and their hosts and that sort of thing. What does this receptor receive? What's the other thing? And hopefully you can predict it based upon really good chemical models. So here is a prediction. Detailed microbial and microbial ecological models will be aided by massive computing power. And computing power, if it follows Moore's law, which I can't imagine it can indefinitely, you know, for 50 years, I think that would mean the transistors would be like smaller than a quark or something. Anyway, I still think the computing power will go way past our wildest imagination, and it will be astounding, okay? Mostly I'm worried that if we'll be here 100 years from now, as Ginger was talking about, I have serious doubts that society will look like it does today, but I think these models will allow us to do things, or these computers will allow us to do things like individually based model, where every cell has its own computer running just what the cell does, everything going on in the cell, if computers get as powerful as they might get, and therefore each individual will be modeled and moved around. So I think that might be something that we can do. And it ranges from viruses up to small animals, as in Terra. Now, as said, it's probably beyond my current imagination, and since it's the end of the day and I thought I'd inject a little levity, I think if I could look ahead into the future, I probably would react the way this guy reacted when he heard about the future. Oh, excuse me.
No, this sucker's electrical. But I need a nuclear reaction to generate the 1.21 gigawatts of electricity.
What did I just say?
The flux capacitor stores. This sucker's electrical. But I need a nuclear reaction to generate the 1.21 gigawatts of electricity.
1.21 gigawatts! 1.21 gigawatts! Great Scott! What the hell is a gigawatt?
Yeah, I have a feeling that the computers of the future will be completely incomprehensible to us today. 1.21 gigawatts was what he needed to run his time machine. And this was the 1955 doc looking at the 1985 doc. And later on, I didn't include this part, but then the kid says, well, all we need is some plutonium. And Doc says, oh, I'm sure in 1985 you can get plutonium in any corner drugstore, but here in 1955 you can't. Okay, so our ideas about the future are usually pretty amazingly off, what we think might happen. But I still think it'll be, I think computing powers will be, that's what we have found in terms of past predicting to the future. We were not able to predict how good computers are. That is really the big change. We aren't all flying around in jets and cars and going to Mars every day or anything like that, like people said, but computing is doing better than anyone ever thought. Way, way ahead, and I think our field will benefit greatly from that as well. Now, predicting more on like a 20-year horizon, I think this is my hopeful thought, that people, the pendulum to only looking at genes and omics and stuff will move a little bit back, so people will realize, yes, we need to go back and measure rates more, we need to measure biomass more, we need to actually look at stuff, we need to measure the respiration and uptake, et cetera, because just looking at omics doesn't give us that information, and we still need it to study ecology. And while it's terribly exciting and has been kind of faddish in some ways to do just genetic work or proteomics work, I think it has to become more balanced, and I hope the information isn't lost as most of us retire who know how to do some of this stuff. Anyway, so another problem is old-fashioned plankton identification requires expertise, and very few young taxonomists are being trained. And even transition to purely molecular studies means someone needs to identify the thing that you put into the database. So if you don't have someone who could identify this diatom or whatever it is, and then you sequence the DNA, who cares if you said, well, it was an interesting unidentified diatom that had this DNA sequence, okay? So you need those folks to be able to do that. And we know that it probably is being done. We heard that there are ways that this stuff is being done. And I think that just as Lee talked about earlier, as having the flow cytobot that recognizes phytoplankton, I think within 20 years that there will be databases and software so that anyone will whip out their phone microscope and take a picture of an interesting little metazoan or a diatom or something, and it will instantly go back to their Facebook-ish database, and it will be recorded that this Chaetoceros gracilis was found at this exact location and time by this person. Okay, and that stuff can all be centralized, and that will be really neat. And I think that will probably happen. It will democratize our field pretty well. You won't have to go and send off your sample to the expert who knows what something is. Hopefully it'll be popular. There's always people that are interested in science even though we don't know them. And I think it'll greatly improve our ability for at least recognizable things to be able to tell what they are. I put this up here. Does anyone know what this is, by the way? I'm curious. We have a whole room full of experts. It's one of these things, you know, I took this picture years ago. No one has ever been able to tell me what it is. It's about 10 microns across. Anyway. But it's one of those, oh, I don't know. Okay. There's a lot of those things out there. Okay. So here's another prediction. Looking 20 to 50 years into the future, sequencing will be dirt cheap, so every kid will get stuff sequenced as part of their science fair project. Imaging will be really cheap. Databases will be huge and accessible instantly by our pocket computers. We don't just have access to more and better microbial ecology, but in fact, I think it could get more interest of the public. More citizens projects, more elementary school. This little girl, instead of looking at bread mold in these plastic bags, she will have the complete genetic analysis and species lists and all the rates and everything else of the bread mold, and where she's, 7 or whatever. I hope that that's what happens in the field, because I think that would be really fantastic. Okay? And I just want to acknowledge for the data that I showed it's the work of a lot of different people. Support's been through the Moore Foundation and NSF, and I'm ready for questions.
But surely no matter how you look at it, you're going to need somebody in the lab who's going to go and knock on those unknown genes and just crack them until they figure out their functions. There's no way a protein sequence will tell you what they do unless somebody in a lab has done some wet experiments to figure it out. Do you agree?
I think for many that will be the case, but for things like affinities and rates, if you already know something is, say, an ammonium transporter, I predict, and I'd be completely wrong, that 50 years from now, a protein chemist can say, this particular one, because it has a valine here and an arginine here, it actually, the hinge is faster, and it can have a higher rate. Or see this one here, this binds the ammonia a little tighter, so it slows down the rate, but it has a higher affinity. I think that for a lot of existing proteins, rates and kinetics and affinities will be known. And some things we'll have to just crack in the lab. Some things we'll never understand, probably. But I have a feeling we'll at least be able to look at a protein and say, the substrate has this exact diameter and this charge distribution, and it looks like it oxidizes it or reduces it or adds a proton to it or adds this to it or combines this or that or breaks a bond or whatever. I have a feeling a lot of those things will be predictable, and it might at least point you to what you can study in the laboratory, rather than working blindly, which we'll do with a lot of proteins today. But you're right, it's never will solve everything. Mike.
You talked about the importance of rates. You talked about I'll bring up rates again. So we're using methods and rate measurements actually made in the 80s. Are there going to be ways to automate those?
Well, some of as you know, there's a lot of optical measurements that people are using for photosynthetic properties, and often you could approximate rates with those. We have global maps of primary productivity from satellite. Now, I know if you look at the error bars that we saw in Emmanuel's diagrams, that there might be an order of magnitude, if you actually look at those things in between the field measurements and the satellite, but they have such huge coverage and things tend to average out. Whether you can use that on an individual sample is a really good question. And some of them are semi-automated, but there's going to be a time that it's going to be hard to buy tritiated substrates. And stable isotopes are always going to be complicated to work with because you need a mass spec to analyze them. Although, if you think about it, back in the days of the Viking lander on Mars, there was a mass spec the size of a coffee cup that was sent there. And people can make really small instruments if they want them and if they want to use them for certain applications. So you could probably put them on a mooring if someone spends the money to design them and put them in. So I think it's possible, but it depends on investments. And some isotopes are going to continue to get used for medical purposes because they're used for imaging and stuff. And those, at least, there's a lot of short-lived isotopes and stuff will always be available for a while anyway. Okay.
And Jed, what do you think about the frequency of sampling and the Lagrangian sampling of the ocean? Or how do you think these should?
I think it's important to have all frequencies of sampling.
Lagrangian?
At least they should. And Lagrangian is valuable. But I think we also have to realize, from the oceanography perspective, that if you ever think you could follow a parcel of water, you're fooling yourself. Because if you ever put down, let's say you follow a drogue, and then make the mistake of putting down two drogues, guess what? They go in two different directions. They're 2 miles apart in a day. Said, which one should I follow? Well, neither one. That's how the ocean goes. It mixes and changes. So Lagrangian, I think it has a concept. It's an idea that you follow a parcel of water. In some ways, parcels of water kind of exist. But I think from the perspective of microbes, a true exact parcel doesn't. But a region that has a coherent set of conditions that certain microbes live in, that's okay. In our studies, they tend to be like kilometers or tens of kilometers in horizontal extent. But as you know, in vertical extent, things change in meters. So there's all that, the 3 dimensions. Then there's internal wave stuff, which complicate a lot of our deep sea measurements. Even, not so deep, even 20 meters. You could sample at 20 meters now, and then wait 20 minutes and go to 20 meters. The temperature is 1 degree different. It's not the same water anymore because internal waves are moving up and down. So oceanographers have to inform you, don't sample at the depth, sample at the temperature, which very few people here think about, but that's, if you want to follow the same water, at least you can track temperature. There's something like that. So there's clever ways you can use oceanographic data to improve tracking things, but it never really tracks.